Cyclic electron flow. A second electron transport pathway called cyclic electron flow produces ATP without the synthesis of NADPH, thereby supplying additional ATP for other metabolic processes. In cyclic electron flow, light energy harvested at photosystem 1 is used for ATP synthesis rather than NADPH synthesis as depicted in the figure. Instead of being transferred to NADP+, high energy electrons from photosystem 1 are transferred to the cytochrome Bf complex. Electron transfer through the cytochrome Bf complex is then coupled, as in photosystem 2, to the establishment of a proton gradient across the thylakoid membrane. Plastocyanin then returns these electrons to photosystem 1 in a lower energy state, completing a cycle of electron transport in which light energy harvested at photosystem 1 is used to pump protons at the cytochrome Bf complex. Electron transfer from photosystem 1 can thus generate either ATP or NADPH depending on the metabolic needs of the cell. ATP synthesis. The ATP synthase of the thylakoid membrane is similar to the mitochondrial enzyme. However, the energy stored in the proton gradient across the thylakoid membrane, in contrast to the inner mitochondrial membrane, is almost entirely chemical in nature. This is because the thylakoid membrane, although impermeable to protons, differs from the inner mitochondrial membrane in being permeable to other ions, particularly magnesium and chloride. The free passage of these ions neutralizes the voltage component of the proton gradient. So, the energy derived from photosynthesis is conserved mainly as a difference in proton concentration across the thylakoid membrane. However, because the thylakoid lumen is a closed compartment, this difference in proton concentration can be quite large, corresponding to a differential of more than 3 pH units between the stroma and the thylakoid lumen. Because of the magnitude of this pH differential, the total free energy stored across the thylakoid membrane is similar to that stored across the inner mitochondrial membrane. For each pair of electrons transported, 2 protons are transferred across the thylakoid membrane at photosystem 2 and 2 to 4 protons at the cytochrome Bf complex. Since 4 protons are needed to drive the synthesis of one molecule of ATP, passage of each pair of electrons through photosystems 1 and 2 by non-cyclic electron flow yields between 1 and 1.5 ATP molecules. Cyclic electron flow has a lower yield corresponding to between 0.5 and 1 ATP molecule per pair of electrons.